Huh?
How to adjust the flow rate? Today we adjusted 300.
How do I adjust?
What is the logic behind
Logic
that?
is very simple. That's what I was saying. Suppose there is a leaf which is transferring very quickly. Then the humidity built up inside the chamber will be very fast. How do I flush it out?
Through flow rate.
Increase the flow rate.
It is simple. You understand this. बहुत ज़्यादा मॉइस्चर है, करने से सूखेगा नहीं ना, ज़्यादा हवा डालने से सूख जाएगा. So if you increase the flow rate, the drying will be fast. But then you should not increase it to such an extent. There will be total.
You cannot make it more than 100% anyway.
So
what will be the optimum flow rate? Like
500 is the maximum that you can.
Maximum.
200 is the minimum that you can. Between this range. If the relative humidity is not alterable by your flow rate, then you go to the scrub business. You reduce the input itself by allowing a part of the air to go through the desiccant.
When it passes through the desiccator, there itself the moisture is reduced.
This is something which you should remember, so that if there is any problem, you can troubleshoot it. A lot of people have done a lot of different kind of things. What you have to do is, you know that these are all tubular connections. You connect a tube to that, put it into some place, and then suppose you want to measure all these things in, let's say sometime in winter when the humidity outside is very, very less, atmosphere is very dry, December, November and all that will be very dry, right?
Not that dry, not here.
But in January in Bangalore, your lips are all cracked because
but here it is not that bad.
Suppose you have a very dry condition outside, and then your air in is, let's say, 35%, 40% humidity.
That happens in summer months for us.
Suppose whichever month.
April, May, June, May.
April, May and all that. If it happens like that, dry conditions, then your growth chamber, the plants are growing at very beautiful 70% RH. You keep them out, they will immediately die in that condition.
Yeah.
So, today also yesterday we saw that we kept the plants and they started drying because of the VPD, temperature and all those kind of things, besides humidity. But in summer, humidity is less, temperatures are high, the plants will very quickly evaporate and die. So if you want to avoid that, what you should do, there is a humidifier also. You don't have it in this.
Okay.
There is a mechanism where there is a mat where it will suck atmospheric, it will add moisture to the system.
Okay.
Suppose you did not have what to do, nothing to worry. You know what I used to do? You take a flask, the round conical flask. You make 2 tubes, and this air, that one is that throw it out somewhere. Allow that one end to get into the water, other one upstairs here. So when it bubble it through, it will capture moisture and then go inside. Easy. You can do, the reason why I am saying that is, you can do any kind of addition to the system if you know what you want. These are extremely simple systems. There is nothing called, there is no rocket science in it. You do molecular biology, therefore this must be very simple. But you remember, what is it that the plant should be given for the best response? It's something like this. That's why I have been telling you this since yesterday. You have a dream, and if you have to tell me whatever dream that you have, I should give you the most appropriate condition around you to know your responses. It's as simple as that. If you want to give the most appropriate condition to another person to find the responses, plants are very beautiful organisms. If you give them, they will never tell lies. Human beings, we tell a lot of lies. We don't. The plants, they learn. And that is what you should do. And once you measure it, and then this is only gas exchange. It's just that at this CO2, at this light, at this humidity, this is what it is. But you can do many, many things. Alter it. You don't know, you do not have a mechanism to alter the CO2 concentration in this system. But I think Madan Pal's system may have that. But if you can alter the light intensity from 0 to 2000, every 100 ppm, 100 micromole light intensity that you change, you measure photosynthetic, measure gas exchange, everything, log. Take the data out and plot. What is the relationship that the photosynthetic rate has on the y-axis with respect to the increase in light intensity on the x-axis? What is the response the photosynthetic rate has with increasing concentration of CO2 on the x-axis, the VPD on the x-axis changes. This will give you a lot of very beautiful information. The initial slope of every response curve is an indicator, like for example CO2 response curve. The initial slope of the CO2 response curve is a very good indicator of carboxylation efficiency.
Okay.
And the initial slope of the light response curve is a very good indicator of quantum efficiency. So this is something which are non-invasive, very beautiful. And of course, the moment you have those kind of things, you can go to fluorescence mechanisms and measure what are the fluorescence kinetics and those kind of things. That is a little more specialized. We can discuss another time. So that's it. Any other question?
No questions? Yeah?
Fluorescent quantum one option is there.
आगे आ जाओ. Somebody is asking question.
What is that?
Fluorescent quantum one option is there. When we measure that fluorescence
Yeah.
shape,
If we have GFP plant means we can measure.
Oh, that kind of fluorescence. No. This is your green. See, the intensity of the fluorescence that you talk about in the GFP and all those things is so low that these systems cannot see that. These are all hardcore things. What comes from photosynthesis is very huge intensities of radiation. But the philosophy behind that is, for example, your green fluorescence protein that you have will give you a green fluorescent light when it is excited with some other light. Isn't it?
When it is excited under UV light.
Under UV it will give you, but there is no UV at all in this. This is all white light.
So you cannot detect that kind of fluorescence.
That kind of fluorescence you cannot detect. But what is the fluorescence that we are detecting here is that I get energy. For example, chlorophyll, that is what you are measuring. Chlorophyll absorbs red light best. Blue light also it will absorb. But this absorption of the radiation will excite the molecule to an excited state. Every electron, the outermost shell electron will get excited and go to the excited orbit. It cannot stay there for long. It will come down to the ground state. During that traveling back to the ground state, the time that takes is 10 raised to minus 12 to 10 raised to minus 9 seconds. Very quick. This fluorescence happens instantaneously. It goes up and comes down. But coming down, it emits the energy either in the form of heat or in the form of light. The light that we see here is not your green fluorescence. Basically, remember this, what happens is, any fluorescence is coming because of excitation of a molecule by light energy, photons only. Fluorescence is the light, and your whatever that has to excite it also must be light. But fluorescence emission spectrum will be longer wavelength than its absorption spectrum.
Fluorescence emission spectrum is longer than the absorption spectrum.
Yeah. Green radiation, green fluorescent protein, which will give you green light when it is excited, cannot give you green fluorescence when it is excited with red light. Anything weaker wavelength cannot give you fluorescence. Stronger wavelength only, stronger wavelength is
the UV,
more energetic radiation.
energetic.
UV is something which you excite, and then gives you fluorescence. This fluorescence is from an excitation energy coming from red light.
Oh.
So 680 is the absorption peak of chlorophyll.
That's why it's called
Emission spectrum is
infrared.
720. Emission spectrum is 720 ppm. 720 nanometers.
what is the exact time, sir, when you are taking readings? Sir, morning.
Very good. Oh, that's a good question. Basically, in a natural condition.
I think you should repeat the question.
Yeah, you repeat the question.
Repeat the question.
So, morning we take the observation and the carbon concentration is 420 ppm. And that time photosynthetic rate is around 7.5.
Okay.
But when we take afternoon, the carbon concentration is 390 ppm.
Yeah.
And that time the photosynthetic rate is
higher. Yeah, very good.
Up to 10.
Very good. Nice. That's a good question. So, what is the
Which
time that
we
we
have to
should
take
take?
observation?
The most typical time that we should record photosynthesis is when the photosynthesis is at its best. There is no point in making you feel very, very sad late in the afternoon and ask you to do a mathematical equation, and when I ask Divya, she is in a happy mood, and then tell that she is a better person. No chance. That is not correct. That is not at all correct. Now, what we do is the best time is between 9 o'clock in the morning and noon. Typically, tropical species, they go through what is called as midday depression in photosynthesis. There is an increase, they stay there for a certain time, and at 12 noon or where little between 1 o'clock and 2 o'clock, because the temperatures increase and peak at 2 o'clock. Humidity will be lowest at around that time. Because of the dynamism between temperature and humidity in the air, VPD will change drastically. So the VPD responses by the plant is very strong. So there is the decrease. At midday, what happens generally is evaporation of water will be generally more than absorption of water.
Because of the temperature.
Because of the conditions, means the temperature and the humidity outside.
Okay.
Both of them. That's why we say VPD, putting them together.
Yes.
Vapor pressure deficits. Therefore, the best time is between this. But you kept on measuring it till 4 o'clock because the plants that you are using are those guys who do not know variations. They are that Siddhartha who became Gautama Buddha later, who did not know anything. He was happy in a temple, what is that, palace. So these guys were protected inside. They are given same amount of light intensity for 16 hours as day and same amount of no light at all, darkness for 8 hours the remaining time of a day. So they are in a very specific rhythm. The relative humidity is 70% all the time, light intensity is exactly the same all the 16 hours. So it doesn't know fluctuations, but outside it's not like that. When you go out...
So then how to interpret the data? So
good, that's very nice.
So when we are putting in growth chamber, they are in control condition.
This is growth chamber only.
Field, there is a
we are
different...
not talking anything about the field at all now, right?
But when we are relating...
When you go to the field, go and measure it in the field conditions. What generally people do is, they will measure diurnal fluctuations in gas exchange. Start at 6 o'clock or 7 o'clock in the morning, go till 6 o'clock or 7 o'clock in the evening. Go measure, monitor what happens, see what happens. You will be very, very happy to see that everything increases. By 7 o'clock, 8 o'clock, everything peaks, stay there till 12 o'clock, and after that they come down. By 2 o'clock, they have already shut down completely, sleep, go to bed, finished. They take leave, that is a diurnal pattern. You don't have diurnal. See, the question is, what is it that you want to interpret? What is it that you want to say? Do you want to say that the brinjal plant that you measure today will exactly behave in the field like they were behaving in the growth chamber? No, that's not your objective. Your objective is very, very simple. Prakash, let's be very clear. Our objective is not to find out what is the relevance of photosynthesis. Our objective is to find out what happens to photosynthesis because of your infection. That's our objective.
Shall I ask you a question? Like now, we did not find any significant differences.
Yeah, correct.
Could it be because both the stressed and the non-stressed plants, they were kept under very ambient conditions?
No, I don't know, ma'am, because I have a feeling we do not know whether the stress is really stress at all.
Okay.
That is the first question.
Okay.
If there is real stress, you can try this. You can simply try this another day. What is that? I mean, I do not know whether that nematode is eating at all or not. What you do is keep one plant, don't water it for 2 days with stress. You keep them in the same 70% RH and same amount of light, same time, and then you measure those guys, you will see there is a lot of difference. Stress has not been influencing the plant for its changing metabolism. That's what I feel, number one. Number two is this, what you said is also possible. But then the question is, if I keep all the plants in a cozy environment, then the stress effect does not come in.
Exactly.
Then the stressor, that nematode or water stress or whatever, is not really a stress at all. It is because of the environment in which if we can keep the
it's an interaction with an environment that makes a plant feel stressed out.
Absolutely correct. But any environment, in a given environment, these fellows must create some stress. Some stress. I feel that it has not been enough.
The stress is not enough to be felt by the plant.
Yes, it is not.
So that it can show that I am suffering.
Yeah. Therefore, that is what I was thinking. Since it looks like that, you should, you may perhaps want to measure the leaf-water relations.
That will be a good idea.
We should do that.
Yes.
And if you show, I don't know if there is any information which is clear in the literature which says this is how the nematode infects and this is what the primary cause of infection is this. The consequences are many. There are 2-3 things, What is the direct effect of a stress and what is the indirect effect of that on other metabolic activities. So most of the time we only measure the indirect effects.
So it affects the oxygen pathway.
It does affect the oxygen pathway. Then it will be very, very nice to find out what happens if the oxygen pathways are not proper. Root growth will not be proper, or the stem elongation will not be proper, leaf expansion will not be proper. Therefore, the calorie photosynthesis will not be proper. But with auxins, there may not be any change in per unit leaf area photosynthesis. We are only measuring unit leaf area photosynthesis,
So
not the whole thing. So you can look at all those things. Look at the total area, Look at the entire thing as a system. You know, then only things will start becoming clear.
Anything else? Vivian? Okay.
Thank you.
So what if
Same.
we take CAM plants or is there any, what will be the criteria if we take CAM plant or C4?
C4 and C3 plants are very similar to what we are looking for. Now they are looking at brinjal, and you looked at amaranthus, which is a dicot similar to this. It would exactly be the same thing, but the efficiencies which those guys do things, C4 plants are much superior. CAM plants are a very different fellows. These CAM plants have temporal variations in photosynthesis. That is, there are two carboxylation that happens in C4 and CAM plants. One is driven by the PP carboxylase, another one is driven by the RUBP carboxylase. These guys are spatially separated in C4 plants. They are different in space. One takes place in the mesophyll cell, other one in the bundle sheath cell. We know this. Whereas the CAM plants are temporally separated, time-wise. One happens in the day, other one takes place in the night time. No, but it's just that mechanism which has evolved to make them to survive under extreme arid conditions, xeric conditions. You know, they are called the xerophytes, the CAM plants. If you see the CAM plants all of them are succulent. Cactus, for example, they're succulent. Now they grow in dirtiest of condition, they're succulent. That is because they don't transpire anything at all. They don't lose any water. Therefore, you can see that their growth rates are also very, very slow. You will not keep a maize plant in your house as an ornamental plant. You will keep a lot of cacti, you know, you will derive pleasure. Look, I have so much of cacti that I collected from various places. You know why? Because they don't grow. If they grew, you will throw them.
I had one cactus fruit in Australia. It's called something. It had such a vibrant red color, blood red color.
And you will be very delicious.
It was sort of little sweetish, bland taste.
I see.
But it was really very
but there
vibrant.
is one CAM plant which you all eat. You will love it. Pineapple.
Yeah, it is.
It's a CAM plant. But these CAM plants are very intelligent fellows. CAM plants have derived this mechanism to survive extreme xeric conditions, desert conditions. But if you give them water, they are not in the ordinary C3 plants. CAM plants are very beautiful. They can switch over.
Very flexible.
That's why the CAM plants are also called facultative C3 species.
Okay. So depending on the conditions, they can
If
either
you give
behave
water,
as...
that's why cactus does not grow because it is a CAM plant. CAM plants have constraint. Their carbon assimilation depends on how much of photosynthetic carbon assimilation was possible by the PP carboxylase in the daytime. But the real photosynthesis takes place in them in the night time, in the CAM plants. That is, RUBP carboxylase functions only in the night. In the day, but carbon dioxide does not come from atmosphere. The stomata are closed. The carbon dioxide is generate from the previous night PP carboxylase activity. They would have kept all the carbon dioxide fixed in an acid, or generally malic acid. Then malate is taken out of the vacuole in the CAM plants and decarboxylated, and the carbon dioxide is fixed by the RUBP carbon. This happens inside closed stomata situation. If the stomata open, photosynthetic rate can become very high. In CAM plant, the C3, the RUBP carboxylase will finish up everything by 10 o'clock and then they go to rest. So why? You can extend that. Pineapple, if you provide water, it will open the stomata during daytime, more carbon dioxide will come, these guys will grow.
They're beautiful, smart.
What is that? Oh,
Plants
CAM
are
plants are very smart.
very
Plants are very, very smart. Cells are very smart. There is a saying, man is kind, but men are cruel. But every cell is very beautiful, very intelligent. The cell doesn't have anything which is waste. But tissue are stupid. But you make the organism which worse. Human being is the worst. Because you have other things, like good thoughts, ugly thoughts. Plants have only one thought, survive. So I'll eat, survive.
Do plants have feelings?
I don't know. They have. I believe they have. Because you go talk to them, plants, my dear plant, sweetie plant, very nice you are. You give me some good data, that fellow gives. Do that, yeah, please. There are a lot of such stories, where they were singing songs with it, and then whatever
Not
it is.
singing songs, but take care. Take care very nice.
Yeah, you take care. You it's like echo, you take care of me, I'll take care of you. Simple. Logic is very, very straightforward and simple. That's what I said. You have a dream that you want to get it, it will come to you. You try this. You try empty. I very strongly believe this. I wanted to read this Paulo Coelho's book, The Alchemist. You read that? If you want to achieve something from the bottom of your heart, the entire universe will conspire you to achieve it.
Will conspire or help you?
Help you. Conspire to help you achieve it.
Okay.
The whole thing, devils, gods, everybody will come together.
Good and bad.
Copied in one movie.
Shah
Rukh Khan's movie. Yeah.
Shah Rukh Khan dialogue.
So that of course must be there. The authors also copy one another, but write it in different languages. It's all there in Upanishad and all that. Anyway.
I was